TCA cycle, also known as Krebs cycle, involves the oxidation of acetyl-CoA to CO2 and H2O. It is also the central metabolic pathway and the final common oxidative pathway for carbohydrates, fats, and amino acids. It also supplies energy and provides many intermediates required for the synthesis of amino acids, glucose, and etc. The TCA is the most important central pathway connecting almost all the individual metabolic pathways. It occurs in the mitochondrial matrix in the close proximity to the ETC. Reaction of TCA cycles. Oxidative decarboxylation of pyruvate to acetyl-CoA by PDH complex. This is the step that connects the link between glycolysis and TCA cycle. Here are the reactions of TCA cycle. Step 1. Formation of citrate. Oxaloacetate condenses with acetyl-CoA to form citrate, catalyzed by the enzyme citrate synthase. It is inhibited by ATP, NADH citrate, a competitive inhibitor of oxaloacetate. For steps 2 and 3, citrate is isomerized to isocitrate. Citrate is isomerized to isocitrate by the enzyme aconitase. This is achieved in two-stage reaction of dehydration followed by hydration through the formation of an intermediate cis-aconiase. For steps 4 and 5, formation of alpha-ketoglutarate. Isocitrate dehydrogenase catalyzes the conversion of oxidative decarboxylation of isocitrate to oxalosuccinate and then to alpha-ketoglutarate. The formation of NADH and the liberation of carbon dioxide occur at this stage. Stimulated by the isocitrate NAD+, Mg2+, AD, Ca2+, links with the contraction. It is inhibited by NADH and ATP. For step 6, conversion of alpha-ketoglutarate to succinyl-CoA occurs through oxidative decarboxylation catalyzed by alpha-ketoglutarate dehydrogenase complex. Alpha-ketoglutarate dehydrogenase is a multi-enzyme complex. At this stage of the TCA cycle, second NADH is produced and the second carbon dioxide is liberated. Step 7. Formation of succinate. Succinyl-CoA is converted to succinate by succinate thiokinase. This reaction is coupled with the phosphorylation of GDP to GTP. This is a substrate level of phosphorylation. GTP is converted to ATP by the enzyme nucleoside diphosphate kinase. For step 8, conversion of succinate to fumarate. Succinate is oxidized by succinate dehydrogenase to fumarate. This reaction results in the production of FADH2. For step 9, formation of malate. The enzyme fumarase catalyzes the conversion of fumarate to malate with the addition of H2O. For step 10, conversion of malate to oxaloacetate. Malate is then oxidized to oxaloacetate by malate dehydrogenase. The third and final synthesis of NADH occurs at this stage. This oxaloacetate is regenerated, which can combine with another molecule of acetyl-CoA and continue the cycle. The regeneration of oxaloacetate. The TCA basically involves the oxidation of acetyl-CoA to CO2 with the simultaneous regeneration of oxaloacetate. There is no net consumption of oxaloacetate or any other intermediate in the cycle. The significance of the tribocarboxylic acid cycle is to complete the oxidation of acetyl-CoA, the ATP generation, final common oxidative pathway, integration of major metabolic pathways. Fat is burned on the wick of carbohydrates. Excess carbohydrates are converted as neutral fat. No net synthesis of carbohydrates from fat. carbon skeleton of amino acids finally enter the TCA cycle. There is no direct participation of oxygen in the TCA cycle. It operates under aerobic conditions. This is due to NAD plus and FAD required for the operation of the cycle can be regenerated in the respiratory chain only in the presence of oxygen. Therefore, citric acid cycle is strictly aerobic. The oxidation of 3-NADH by ETC coupled with oxidative phosphorylation results in the synthesis of 9-ATP. FADH2 leads to the formation of 2-ATP. Once substrate level phosphorylation, thus a total of 12 ATP are produced from one acetyl-CoA. The 3 enzyme that regulates the tricarboxylic acid cycle is the citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. Citrate synthase is inhibited by ATP, NADH, and acyl-CoA and succinyl-CoA. Isocitrate dehydrogenase is activated by ADP and inhibited by ATP and NADH. Alpha-ketoglutarate dehydrogenase is inhibited by succinyl-CoA and NADH. Availability of ADP is very important for TCA cycle to proceed.